Here you can see the equation for cell respiration, and this is an aerobic equation. So you have 6O2, so that would be oxygen gas, then you have glucose, and it's going to produce carbon dioxide, water, and energy in the form of ATP. So remember, the whole point of cellular respiration is that the cells are going to use the glucose, or the carbohydrate, the main source of energy for our cells, and it's going to turn it into the usable form of energy, which is known as ATP, or adenosine triphosphate. Also, recall from previous lectures that there's two types of cellular respiration. There's aerobic respiration when oxygen is present, and anaerobic respiration without oxygen. So remember, with anaerobic respiration, if the cell does not have oxygen, after glycolysis, the cell will go through what's called fermentation. So depending on what type of cell is going to depend on what type of fermentation that cell is going to go through. So for example, our animal cells will go through lactic acid fermentation. Now, if our cells have oxygen, which happens the majority of the time our cells have oxygen, after glycolysis, that pyruvic acid that's made in glycolysis will move on to another step called the Krebs cycle. So here you can see the step one. So regardless if we're talking about aerobic or anaerobic respiration, each respiration always starts with the process of glycolysis. So remember, what's happening is glucose is starting to break down to release the energy as ATP. Now glucose goes into glycolysis and it breaks down into 2,3 carbons called pyruvate or pyruvic acid. In this process, we net 2 ATP molecules, and we also produce a high energy molecule known as NADH, specifically 2 of them. The NADHs that are produced here are going to go right away to the third step of aerobic respiration, which is called the electron transport chain. The ATPs that are produced are used by the cell right away to do any type of work. For example, we talked about active transport, requiring energy to move things from low concentration to high concentration. Glycolysis takes place in the cytoplasm, outside of the mitochondria, and then step 2, we went over the Krebs cycle, that then took place in what's called the matrix of the mitochondria. Here you can see the diagram of the mitochondria structure. So remember, the matrix part is that inner part. It almost looks like the lazy river. Okay, it's full of liquid. And after the Krebs cycle, the next step or final step of aerobic respiration is known as the electron transport chain, or you'll hear me refer to it as the ETC. So that takes place in the inner membrane, and specifically the inner membrane folds, which they call the cristae. In this diagram, you can see the glucose is broken down into 2 pyruvic acids, so that's glycolysis, and now that we have oxygen present, those pyruvic acids are moving into the mitochondria, where they first turn into what's called acetyl-CoA, and then that molecule enters into the Krebs cycle. So Krebs cycle is step 2 of aerobic respiration. Last lecture, we talked about the Krebs cycle, so just to review, the Krebs cycle is also known as the citric acid cycle because citric acid is the first compound that's produced in the cycle. Remember, it's a 6 carbons, so what you're going to see in the diagram is the 2 carbons from acetyl-CoA are going to combine with a 4 carbon known as oxaloacetate, and that becomes citric acid. Now, in the process of the Krebs cycle or citric acid cycle, you're going to see carbon dioxide is going to be produced. So as we're breaking down these carbon molecules, these sugar molecules, carbon's going to be released as carbon dioxide. So carbon dioxide is a waste product. You're also going to see NADH and FADH2 produced. So this is the first time that we're seeing FADH2 produced. We saw NADH made in glycolysis. So more NADH and FADH2s are made. And in the process, we're making 2 ATP molecules. So those 2 ATP molecules are going to be used by the cell right away. It's 1 per turn. So remember we talked about last lecture about how the Krebs cycle, in order to break down the original glucose in the beginning of glycolysis, it must go around the Krebs cycle twice, 1 per turn for each pyruvic acid that we start with. So here's the diagram of the Krebs cycle or the citric acid cycle. We're going to be practicing identifying inputs and outputs. So if it's an input, you're going to see an arrow going in, and if it's an output, you're going to see the arrows are going to be coming out of the cycle. So here are the totals for the 2 turns. You guys don't have to remember these, just remember what molecules are produced or made. So carbon dioxide, NADH, FADH2, and then the ATPs. Eventually, we're going to keep track of ATP molecules produced. So what's going to happen is the carbon dioxide is going to be released by the cell as waste, and that's what we eventually exhale. The NADHs here and the FADH2s that are produced, they're moving on to the next step, which we call the electron transport chain, and then the ATPs are used right away by the cell. Step 3, or the final step of aerobic respiration, is called the electron transport chain, or abbreviated ETC. This happens in the inner membrane of the mitochondria, which we specifically call the cristae, so that's the folded part. What you're going to see in the diagram is you're going to see the NADHs that are produced from glycolysis, and also the Krebs cycle are going to be used, and also the FADH2s produced in the Krebs cycle are used. What they're going to do is they're going to drop off their hydrogens and electrons, because remember they were formed by an electron carrier, and those electrons are going to be passed down what's called the electron transport chain. We saw an electron transport chain when we looked at the light-dependent reactions for photosynthesis. So you're going to see electrons moving, and that's going to cause the H pluses to move. So we talked about how they were moving by passive transport, specifically facilitated diffusion. So you're going to see some hydrogens move across the membrane, and that creates a gradient, a concentration gradient. And you're going to then see the H pluses move back through the membrane through that specific type of enzyme that makes ATP. It's called ATP synthase. So again, we saw ATP synthase when we looked at the light-dependent reactions in photosynthesis. So it's the enzyme that makes ATP. So remember, you're going to see ADP add on a phosphate to then become ATP. Oxygen is considered the final electron acceptor. So when the oxygen gets hydrogens and electrons onto it, that's how the water's then made. The end result of the electron transport chain is you're going to produce 34 ATP molecules, and all those ATPs are coming from the energy that's being carried in the NADH and the FADH2. And you're also going to produce water. So here's a diagram of the electron transport chain. So you can see the phospholipid bilayer, so that's labeled as the inner membrane there. So that's going to be the cristae. And then the matrix would be the inner liquid portion where the Krebs cycle is. So the Krebs cycle is producing the NADHs and FADH2s, and they're going right to then the cristae, or inner membrane. So you can see in the diagram here on the left, so bottom left, NADH and FADH2s are going to be dropping off their electrons and also hydrogens. So you can see the hydrogens are going to be pumped to the inner membrane space side. So remember, that's the area between the inner and outer membrane. So the H pluses are diffusing through facilitated diffusion across the membrane. And you also see the electrons being given up. So the electrons are traveling down the electron transport chain. So you can almost think of it as an electron conveyor belt, or even if you go skiing, you know, the ski lifts. So the electrons are traveling between these inner membrane proteins. They're called cytochromes. Now, the hydrogens are then going to start building up, so they're going to have to diffuse back because it'll become high concentration and they'll want to move to the low concentration area. So when the hydrogens move back, they move back through ATP synthase. So you can see that on the right. It's red. So when the H plus goes through that enzyme, it causes the enzyme ATP synthase to spin like a turbine. And that's going to cause enough energy to add a phosphate onto ADP to become ATP. Also, we have water being produced here. So how the water is being made is when the electrons travel down the electron transport chain, they need to go somewhere. So the final electron acceptor is called oxygen. So when oxygen attaches to the electrons and the hydrogens, that's how water's made. So here's something interesting about the electron transport chain, and some of you have maybe read literature on this. So a lot of times they used to use the poison cyanide to kill people, to poison them. And what cyanide does is it actually prevents the electron transport chain from actually taking place. So when somebody ingests it, all of these electron transport chains in the mitochondria are going to stop. So what that means is the electrons will have no place to go, and they essentially will back up in the chain and form like a traffic jam. So then the electrons can never bind to oxygen. So then water's not made, but really the electron movement is what's causing the hydrogens to move. Therefore, the H pluses will never be able to then go through the ATP synthase, and then no ATP will be made, which means the person will end up dying because they can't produce any ATP, and that's the energy source for their cells. So they'll have no energy, and then all the cells will shut down because they have no energy, and then that's how the person will end up dying. So if we look at the total ATP production for aerobic respiration versus anaerobic respiration, you're going to see in aerobic respiration, the total ATP produced from one glucose molecule that we start with back in glycolysis will be 38 ATP molecules. Whereas in anaerobic respiration, we're only going to be able to produce from the one glucose that we start with in glycolysis, we're only going to be able to produce 2 ATP. So for our cells will go through aerobic respiration the majority of the time, and that's much more efficient than anaerobic respiration, because for one glucose it's 38 ATP versus then only 2 ATP in anaerobic respiration. Here's a diagram summarizing all of aerobic respiration. So you can see the first step is glycolysis. Glucose is broken down to pyruvic acid. We produce 2 ATPs, and then NADH is at the top there. Those pyruvic acids then move into the mitochondria and go to step 2, which we call the Krebs cycle. You're going to have NADHs and FADH2s produced, carbon dioxides released, and ATP. And then the final step of aerobic respiration is the electron transport chain. That's where the majority of the ATP is being produced. Now, oxygen is the final electron acceptor, which then creates the water. So the 3 steps of aerobic respiration are glycolysis, Krebs cycle, and then the electron transport chain.